Hello again, and welcome back to continuing our series on renal physiology. This lecture is going to focus on transport mechanisms, and you'll notice I have a theme here of gears that will persist throughout the slides. I think that is very symbolic of the way that transport works in the kidney, because it's like what happens to one ion or molecule or something that's getting reabsorbed or secreted in and out of the kidney. What happens in one impacts others, and then what happens in the proximal tubule will have a downstream effect on what happens later on. And so it's like it's all connected in some fascinating way. Similar to our previous lecture, this is going to be a video series broken up into more digestible pieces of content. We will go over this again in class and lecture, so please utilize as you see fit. And here are the learning objectives for this lecture. We will begin with the proximal tubule. The proximal tubule, as you may have learned in your anatomy class, I'm not sure if they covered it or not, but as you may or may not know, the proximal tubule is actually made up of two subportions that collectively we call the whole PT. But technically, they are made up of a proximal convoluted tubule and a proximal straight tubule. Collectively, we can refer to that as either the early portion or the late portion of the proximal tubule. I will not go into the physiological differences between these two areas of the proximal tubule. It's kind of beyond the scope of what we're going to be talking about today, but I am going to kind of lump it all up into one portion. And I will probably refer to it all as the PT or the proximal tubule. But to be accurate, I'm probably technically referring to the proximal convoluted tubule, as the proximal straight tubule has its own nuances. However, in your reviews, or if you're using some sort of outside resource or question banks or something like that, you might come across these terms, and I didn't want you to be taken by surprise. Most notably about the proximal tubule, all glucose and amino acids should be reabsorbed here. So what that means is at the glomerular filtration barrier, some glucose and some amino acids will be filtered into the nephron. Any ones that are filtered, all of them, all should be reabsorbed in the proximal tubule. Therefore, that means that if you have glucose in the urine, then something has gone wrong. And most other substances that are filtered, most of them are reabsorbed here. And that's why you'll see how abundant the activity in the proximal tubule is. So we generally say 2/3 of almost all the solutes, sodium, potassium, phosphate, calcium, are reabsorbed here. 100% of the glucose and amino acids are reabsorbed here. 50% of urea is reabsorbed here. We'll talk more about urea later on. And then magnesium is a little bit of a special character. Magnesium is more actively reabsorbed elsewhere, but some is reabsorbed here. These are general notes to keep in mind when it comes to proximal tubule reabsorption. I'm going to break this down into a way that I think makes the most sense to understand what is happening. And in order to keep the action of all of the transporters and solutes and everything straight, let's break up the steps of reabsorption into steps in which they move. Over here on the right, we have a histologic image of proximal tubules in cross section. So all of these round circles are all proximal tubules. Okay, here's one, here's another one, and here's another one down here. When we are looking at the proximal tubule, there is a lumen in the middle, and that is what this is in the middle. And that's where that brush border is. So you see all this fuzzy stuff. That's where all those convoluted little brush border elements take place. Okay, and that is part of the apical border of the proximal tubule epithelial cell. So here's an epithelial cell right here, and then here's another epithelial cell right there. You can kind of see them. These are all their nuclei. Nice dark little pink circles here. And then we have the intercellular compartments, and then across the other side of the cell is the basolateral membrane. So the basolateral membrane is the outermost part. So the inside of the cell where the lumen is, this is where the urine or the primitive urine would be, and that didn't write very well. Let me go down here a little bit. This is where the primitive urine would be, and then the peritubular capillaries, or all of the blood vessels, would be here kind of in between all the cells. They just don't show up very good on histologic images. So what that means is when we are reabsorbing stuff, solutes, water, all of that has to make its way from the lumen, cross this border, get across the apical border, get through the intercellular compartment, and then cross the basolateral border, and then make its way back into the capillary. That's why I break this down in these steps over here on the left. In order for reabsorption to occur, stuff has to cross the apical border, navigate through the cell, cross the basolateral border, and then enter the peritubular capillary. All this activity is driven by the power of a continuously ongoing pump, and that is the sodium potassium ATPase, which is designated by this little star symbol, meaning this is always located on the basolateral side of the cell. So the basolateral side, so the outside of the cell. That's why this is covered in stars on the outside. If these were all donuts, then the stars are the powdered sugar on the outside. That is the sodium potassium ATPase. As I mentioned in the previous slide, the proximal tubule handles most of the reabsorption. It is very active and is very energy requiring as a result because of that sodium potassium ATPase. It burns a lot of energy. When it comes to sodium, sodium is usually moving from the lumen or the urine side into the blood as a couple with some other solute. And that's what we're going to get into next. We're next going to begin to look at the movement of individual solutes as they cross from the urine side or the lumen back into the blood side. And that is the process of reabsorption. Do not confuse reabsorption with the process of secretion. I'm only going to briefly mention it here, but secretion is not the same thing as reabsorption. If secretion was going on, secretion means that things move from the blood or the peritubular capillaries, they move in the opposite direction as reabsorption. So blood into the urine, or in this case, the primitive urine, the lumen. So let's look at what that might look like in one of these cross sections. Let's go down here to this one. We haven't drawn on this one yet. So if you have something in the blood, so blood vessels over here, and something's going to get secreted, that means it's going to leave the body, right? That's what secretion means. Secretion means it's got to cross the basolateral membrane, cross the intracellular compartment, cross the apical membrane, and then dump into the lumen of the nephron. Assuming it doesn't get reabsorbed elsewhere, it would just flow on down through the lumen of the nephron and eventually into the bladder. So that is what our secretion is. It is not the same thing as reabsorption. And you could essentially reverse these steps on the left here, and that would account for secretion. We will now begin to discuss some specifics. Going back to our steps, the first step is to get reabsorbed, something has to cross the apical border, meaning it's going to move from the lumen side, or you could say the primitive urine side, which is what this yellow tube on the left graphic is depicting. It's going to cross the cellular compartment and then move towards the blood, which is what this red tube on the left is depicting. In our graphic over here on the right with the histology slide, that means that to get reabsorbed, something is moving from the apical side or the lumen side. It's going to cross the border, get across the cell, and then get reabsorbed across the basolateral side. So we're going to focus first on this apical membrane and what happens there. In the case of sodium, sodium is reabsorbed most often with a buddy. That buddy means that there are special symporters dedicated to sodium plus its buddy that exist on the apical membrane. And that is what is depicted over here on this side. Those special symporters would be channels or symporters that exist on the apical side. Okay, so these are examples. There's a sodium glucose symporter. There's a sodium amino acid symporter, a sodium phosphate symporter. Most often, sodium is reabsorbed with bicarbonate. Now, it's not that there's necessarily a sodium bicarbonate symporter, but that they move together. And I like to think of it as a buddy system, which is what makes sense to me. But you'll learn more about the movement of bicarbonate in acid-base physiology. Regardless, sodium gets in across the apical side of the cell membrane using a buddy system. Now, there is another way, and that is an antiporter, meaning that sodium, for instance, can cross the apical side of the membrane also in exchange for something else, meaning as for every one sodium that gets in, something else gets kicked out, and that can be a hydrogen or other solutes. There's several different kinds. But the main point is, for the most part, to get across the apical membrane, sodium or one of our other solutes is going to be reabsorbed across that membrane, either with a buddy or in exchange for something else. This slide really focused the most on sodium, but all of the other solutes like chloride, magnesium, potassium, all of that is also covered in your DSA. If you remember from our steps, after something crosses the apical border, the next job it has to do is it has to navigate through the intracellular compartment and then cross the basolateral border. The basolateral border is the outside border, right? And that is where the solute would leave the cell and then have access to be reabsorbed into the bloodstream and then circulate through the body again. In the case of sodium, the question then becomes, well, we reabsorb sodium on the last slide. Now it's inside the cell. What happens to it? Well, when it comes to sodium, sodium exits the cell using the sodium-potassium ATPase. And that's what this circle is over here depicting. A few slides ago, I used this big old star-shaped graphic to depict the sodium-potassium ATPase. But that's what this circle is. And I kind of use the same circle over and over again to depict that. So when it comes to sodium, it's pumped out of the cell using the sodium-potassium ATPase, and then it can get back into the bloodstream, which is what's shown here, this little white sodium. All the other solutes, whether it's glucose, amino acid, chloride, potassium, organic acids, all of the other solutes, they pretty much cross the basolateral membrane for the most part using their own specially dedicated transporter. What that means is somewhere over here on the basolateral membrane is a special transporter that, say, for instance, transports inorganic phosphate or phosphate. And then from there, it can cross and get back into the bloodstream. Or maybe chloride, it has its own special channel, and that's what's depicted here. And then it can get reabsorbed. So usually they have all their own little special channels. And if you go back to the DSA, all of those special channels are listed there and outlined in a more easy to read fashion. Finally, we come to the putting it all together slide. This graphic is showing you the proximal tubule. This particular image and graphic author calls it the early proximal tubule. It can also be the proximal convoluted tubule, or we can just call it the proximal tubule. The tubular lumen side is over here, meaning this is the apical border, and this is the basolateral border on this side. So in this case, solutes are going to get reabsorbed from the tubular lumen side, has to cross the apical border, navigate across the cell, and then get out through the basolateral border and back into the blood. And this is putting it all together, all the things we just went over covering. Here you'll see a pattern. On the apical side, you'll notice sodium. Sodium is usually transported into the cell with a buddy. In this case, with sodium, there's a special transporter called the SGLT transporter with amino acid or with some of the other solutes that are reabsorbed. Sodium can also be reabsorbed in exchange for something else, and that in this case is in exchange for hydrogen. For every sodium that comes in, a hydrogen gets pumped out, and that's using a special transporter called the sodium hydrogen exchanger. Again, these are written out in your DSA for you. Sodium, or some of these other solutes that cross the intracellular compartment, they navigate their way through, and then they have to get over and through the basolateral membrane to get back in the blood. In the case of sodium, all these sodiums end up getting pumped out through the sodium-potassium ATPase. So that's how sodium gets out of the cell, and then it gets sucked up into the peritubular capillary. If we are looking at something like glucose, for instance, glucose entered the cell using this sodium glucose transporter, the SGLT, but now glucose gets out of the cell using its own GLUT transporter. And there's a whole bunch of different GLUT transporters. In this case, this particular GLUT transporter is expressed on the basolateral side of the membrane, and it transports glucose out. Amino acid has its own special transporter. Some of these other solutes have their own special solute transporter. And then there is bicarbonate, which again you will cover specifically in your acid-base physiology. That leaves one major thing to talk about in the proximal tubule, and that is water. How does water get reabsorbed? That's what we're going to talk about next. What about water? Well, remember our steps. Water, if it's going to be reabsorbed, needs to move through the apical side of the membrane, cross the intracellular compartment, and then cross the basolateral side of the membrane in order to gain access to the vasculature and re-enter the vasculature in order to circulate throughout the body again. If that is the case, and that is exactly what happens in the case of transcellular movement of water. Transcellular movement of water, by definition, is that it is a pathway by which something is going to move from inside the cell, through the cell, inside of it, and then outside the cell by crossing all of the cell membranes. So it's transcellular, it moves through the cell. And that is in opposition to the paracellular pathway, where something moves between the cells. So there is no crossing of cell membranes involved. When transcellular movement of water occurs, it has to occur via aquaporin channels, and that is what this graphic down here in the middle is depicting. It's literally a pore, and that's why it's called aquaporin, meaning water hole, essentially. And this pore, as you can see in this graphic, it allows for water to move between its two arms. You can see all these nice little happy water molecules moving through the pore and gaining access from one side of the cell membrane to the other side of the cell membrane. So now we were outside the cell, and now we're inside the cell. There are a whole bunch of aquaporin channels in the kidney, and when it comes to proximal tubule, I don't care that you need to know the name of the aquaporin channel, just that it exists, and that's how water moves. There are aquaporin channels that exist on the apical side of the membrane and on the basolateral side of the membrane, because if water is going to get into the cell, it has to get out again. And so it requires aquaporin channels on both sides. It does move passively, meaning this is not an active process. And then when water gets across the basolateral membrane to get into the vasculature, it enters using a concept called bulk flow. Bulk flow is essentially the phenomenon where water is following the solutes. It has to do with Starling forces and the kind of pressure and pull of water following the movement of the solutes that are entering the blood system. Finally, over here in the bottom, I want to highlight an important physiologic concept, and this is in this yellow box down here on the bottom right. And that is when we are in a state of low effective circulating volume, and we could call that a volume contraction. Volume contraction, it's kind of a weird looking V there. When we are in a volume contraction state, that might be we're dehydrated, we have low effective circulating volume for one reason or another, there tends to be an increased reabsorption of a lot of these solutes. And that is solutes being reabsorbed more readily because we're in a volume contracted state. So why is that? Well, that's because solutes are usually coupled with the movement of sodium. Remember, they are buddies. Sodium usually has a buddy when it's reabsorbed in the proximal tubule. And when we're in a volume contracted state, we want to reabsorb more sodium so that we can utilize this phenomenon of bulk flow and reabsorb more water. And that is going to help try to counteract our volume contracted state or reverse it. And then the opposite effect happens as well. If you have a volume expansion for one reason or another, you're fluid overloaded, then you have decreased reabsorption of solutes because you have decreased reabsorption of sodium and all its buddies. Meaning we can conceptualize, say, take calcium, for instance, that in a volume contracted state, you will have an increased reabsorption of calcium because of this phenomenon. And that concludes this section of the learning objectives and LO1. Stay tuned for more videos.